Hello, everyone, and welcome to the lecture number 4 in our Carbohydrate Metabolism series. I am Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. In previous lectures, we mastered glycolysis, its regulation, and the Cori cycle. Today, we enter the mitochondrial powerhouse, the citric acid cycle, also known as Krebs cycle or tricarboxylic acid cycle. This cycle is the final common pathway for the complete oxidation of acetyl-CoA derived from carbohydrates, fats, and many amino acids. It is the central hub of cellular respiration, supplying reducing equivalents like NADH and FADH2 for the ATP production and providing key intermediates for biosynthesis. Understanding the TCA cycle is essential for grasping energy metabolism, mitochondrial disorders, lactic acidosis, and the metabolic reprogramming in cancer cells. So let's study it systematically with the biochemical precision and strong clinical correlation. So let's discuss the fates of the pyruvate. Pyruvate, the end product of glycolysis, has three major metabolic fates that link glycolysis to other pathways. The very first one is the conversion to lactic acid, that is anaerobic conditions or hypoxic tissues. Second one is the conversion to oxaloacetate via pyruvate carboxylase anapleuritic reaction for gluconeogenesis or replenishing TCA intermediates. The third one is the conversion to acetyl-CoA by a pyruvate dehydrogenase complex, that is irreversible aerobic route that feeds the TCA cycle. And for your clinical correlation, at this very point, in thiamine deficiency, beriberi, and alcoholism, along with pyruvate dehydrogenase that is impaired, leading to accumulation of pyruvate and lactate. That leads to accumulation of lactic acid, and that leads to lactic acidosis. So this slide reminds us that pyruvate is the critical junction between glycolysis and the TCA cycle. Now let's discuss the conversion to lactic acid. That is the very first fate of the pyruvate. Under anaerobic conditions or in tissues with high glycolytic flux, for example, in case of RBCs exercising muscle, retina, lens, pyruvate is reduced to lactic acid by lactate dehydrogenase. This reaction regenerates NAD+, allowing glycolysis to continue. So, excessive lactate production causes lactic acidosis, a medical emergency seen in shock, sepsis, severe anemia, and in metformin toxicity. The next fate is the conversion to oxaloacetate. Pyruvate is carboxylated to oxaloacetate by pyruvate carboxylase, that is biotin dependent and activated by acetyl-CoA. This is an anapleuritic filling up reaction that replenishes TCA cycle intermediates and provides substrate for gluconeogenesis. So, pyruvate carboxylase deficiency is a rare but severe inborn error causing lactic acidosis, hypoglycemia, and neurological impairment. The third fate is the conversion to acetyl-CoA. The major aerobic fate, pyruvate is oxidatively decarboxylated to acetyl-CoA by the multi-enzyme pyruvate dehydrogenase complex. This irreversible step commits carbon from glucose to complete oxidation. So, PDH deficiency, pyruvate dehydrogenase complex deficiency, or the PDH deficiency or thiamine deficiency leads to the buildup of pyruvate and lactate, causing congenital lactic acidosis and neurological deficits. Arsenic and heavy metals also inhibit the pyruvate dehydrogenase complex. So now let's discuss the overview of the citric acid cycle. The citric acid cycle consists of 8 enzymatic steps occurring in the mitochondrial matrix. For academic clarity, we divide it into 4 stages. It begins with the entry of 2-carbon acetyl-CoA and ends with the regeneration of oxaloacetate, producing 3 NADH, 1 FADH2, 1 GTP or ATP molecule, and 2 carbon dioxide per acetyl-CoA molecule. Now let's proceed to the stages of the TCA cycle or the citric acid cycle. Stage number 1, that is the formation of citric acid. So citrate synthase, the very first enzyme, acetyl-CoA plus oxaloacetate leads to the formation of citrate. And furthermore, aconitase, the enzyme, converts citrate to cis-aconitase and then to isocitrate molecule. This stage condenses a 2-carbon unit with a 4-carbon unit to form a 6-carbon citrate molecule. Citrate synthase is a key regulatory enzyme. As you can see on your screens, the very first enzyme is the citrate synthase, and the next one is the aconitase that leads to the formation of isocitrate molecule. Now, let's proceed to the stage number 2. That is the formation of alpha-oxaloglutarate molecule. Now, isocitrate dehydrogenase, ICD, the enzyme isocitrate molecule converts to oxalosuccinate, and that leads to the alpha-glutarate, that is oxidative decarboxylation, producing 1 molecule of NADH and very first molecule of carbon dioxide. And then the alpha-ketoglutarate molecule changes to succinyl-CoA. In the presence of alpha-ketoglutarate glutarate dehydrogenase complex. That is another oxidative decarboxylation process, producing another NAD and carbon dioxide. So this stage releases 2 molecules of carbon dioxide and generates 2 NAD molecules. So clinical correlation at this very point is this alpha-ketoglutarate dehydrogenase is structurally and functionally similar to the pyruvate dehydrogenase complex and is also thiamine dependent. So let's move to the stage number 3. That is the conversion of succinyl-CoA to succinate. Succinyl-CoA synthetase molecule, the enzyme, or the succinate thiokinase, converts succinyl-CoA to succinate, producing GTP or the ATP in some tissues by substrate level phosphorylation. This is the only substrate level phosphorylation step in the TCA. So this is a very high yield exam question, right? Or point. As you can see on your screen, the stage number 3, that is the conversion of succinyl-CoA to succinate molecule. Now, let's proceed to the stage number 4. That is the regeneration of oxaloacetate molecule. Succinate dehydrogenase is the enzyme that converts the succinate to fumarate, producing very first molecule of FADH2. And then fumarase convert the fumarate to malate or malic acid. Now, the oxidation of malic acid in the presence of malate dehydrogenase convert that molecule to oxaloacetate. And that is the stage 4, the regeneration of oxaloacetate molecule. And then this completing the cycle and ready to accept another acetyl-CoA for further proceeding. Now let's discuss the energetics of the citric acid cycle. Per molecule of acetyl-CoA, that is 1 turn of the cycle. So 3 NADH produces about 7.5 ATP via ETC. And 1 FADH2 molecule produces 1.5 ATPs. And 1 ATP or GTP molecule is equal to 1 ATP molecule. So total 10 ATP per acetyl-CoA. Since 1 glucose yields 2 acetyl-CoA, the TCA cycle contributes 20 ATPs per glucose molecule plus glycolysis and PDH. Complete aerobic oxidation of glucose yields 30 to 32 ATPs. Furthermore, mitochondrial disorders affecting TCA enzymes and electron transport chain cause severe energy failure and lactic acidosis and multi-system disease. Now, let's proceed to the regulation of the citric acid cycle. The cycle is regulated at 3 key enzymes. The very first one is the citrate synthase, allosterically inhibited by ATP and long-chain acyl-CoA. That is a signals high energy condition. The second one is the isocitrate dehydrogenase, that is activated by ADP and inhibited by ATP and NADH molecule. The third one is the α-Ketoglutarate dehydrogenase, that is regulated similarly to the PDH complex, inhibited by NADH, succinyl-CoA, and the ATP. So, high NADH to NAD plus ratio that occurs in alcohol metabolism or hypoxia shows the TCA cycle diverting pyruvate to lactate formation. So, this is the summary of the TCA cycle showing all the reaction of all 4 stages. We have now comprehensively covered the fate of the pyruvate, the 8 reactions of the citric acid cycle, divided into 4 stages, its energetics and its regulation. The TCA cycle truly stands as the metabolic crossroads of the cell, linking carbohydrate, fat, and protein metabolism while generating the majority of the ATP through oxidative phosphorylation. In the next lecture, we will discuss the electron transport chain, oxidative phosphorylation, and the integration of the metabolism. Thank you very much for your attention and for watching this lecture. If this session strengthened your understanding of the citric acid cycle and its clinical relevance, please like the video, share it with your fellow medical students and colleagues, and subscribe to the channel for the complete high-yield medical biochemistry series. Turn on the notification bell so you never miss upcoming lecture. So drop your questions, doubts, and interesting clinical cases related to the TCA cycle in the comment section. I read and respond to every single comment personally. Which step or regulatory mechanism would you like me to explain in greater detail? Please let me know. This is Dr. Muzamil Liaqat Ali signing off. Revise the 4 stages through regulatory enzymes and the energetics daily. Stay focused, maintain intellectual rigor, and keep mastering biochemistry with passion and precision. I look forward to seeing you in lecture number 5. Thank you once again.